Hello, today we will be covering sex chromosomes and genetic inheritance. So let's talk about sex chromosomes. Sex chromosomes are a pair of chromosomes that determine an individual's sex or gender in many species, including humans. These chromosomes carry genetic information that influence the development of an organism as male or female. In humans, sex chromosomes are denoted as X and Y. So let's talk about chromosome pairs. Humans have 23 pairs of chromosomes, including one pair of sex chromosome. The other 22 are called autosomes. Female chromosomes, like we just said, typically have two X chromosomes, denoted as capital X, capital X. These chromosomes carry genes related to female secondary sexual characteristics and reproductive functions. Male chromosomes usually are denoted by an X and Y. The Y chromosome carries genes responsible for male secondary sexual characteristics. So let's talk about sperm and egg cells. So during reproduction, sperm cells from males carry either an X or a Y chromosome, while egg cells from females always carry an X. The combination of the sperm and egg chromosome determines the genetic sex of the offspring. Now let's talk about inheritance. So a child's sex chromosome combination is inherited from their biological parents. The mother always contributes an X, while the father will always contribute either an X or a Y chromosome to the child. And now let's talk about DSD, disorders of sex development. Occasionally, there can be variations in sex chromosome combinations, leading to conditions known as disorders of sex development. These conditions may result in individuals with atypical chromosomal, gonadal, or anatomical sex characteristics. Let's dive into genetic linkage. Linkage refers to the tendency of certain genes located on the same chromosome to be inherited together during genetic transmission. This concept was first described by British geneticist Thomas Hunt Morgan in the early 20th century through his work with fruit flies. Morgan discovered that genes on the same chromosome tend to be linked because they are physically close to each other on the chromosome and are often inherited together as a unit. However, not all genes on the same chromosome are always linked, as genetic recombination can occur during meiosis, the process of cell division that produces gametes. During recombination, segments of chromosomes can be exchanged between homologous chromosomes in a process called crossing over. This shuffling of genetic material can break the linkage between genes on the same chromosome. Sex chromosomes can also exhibit linkage, although the extent of linkage depends on the specific genes involved and their positions on the sex chromosomes. Some sex-linked traits are carried on the X or Y chromosome and are inherited in sex-dependent manner. For example, hemophilia and color blindness are examples of X-linked recessive disorders, meaning they are more commonly expressed in males because they have only one X chromosome, while females have two X chromosomes and can often carry one healthy allele to compensate for the defective one. So, to summarize, sex chromosomes play a crucial role in determining an individual's sex and carrying genes related to specific traits. While they exhibit linkage, genetic recombination during meiosis can break these linkages, contributing to genetic diversity within populations. Our next slide is over non-disjunction. Non-disjunction is a genetic phenomenon that occurs during cell division, particularly during meiosis, where chromosomes do not separate or segregate correctly. This results in an unequal distribution of chromosomes into the daughter cells, leading to abnormal chromosome numbers in the offspring. Non-disjunction can occur in both mitosis and meiosis, but its consequences are more significant in meiosis. Non-disjunction can occur during the separation of homologous chromosomes in meiosis 1 or the separation of sister chromatids in meiosis 2. So let's talk about how non-disjunction can occur in genetics. So in meiosis 1, homologous chromosomes are supposed to separate into two different daughter cells. Non-disjunction in meiosis 1 occurs when the homologous chromosomes fail to separate. As a result, one daughter cell receives both homologous chromosomes with an extra chromosome, leading to a condition called trisomy. The other daughter cell receives none of the homologous chromosomes, leading to a condition called monosomy. A common example of meiosis 1 non-disjunction is Down syndrome, which results from an extra chromosome, 21. So now let's talk about non-disjunction in meiosis 2. So during meiosis 2, the sister chromatids of each chromosome are supposed to separate and go to separate daughter cells. Non-disjunction in meiosis 2 occurs when sister chromatids fail to separate correctly. As a result, one daughter cell receives both sister chromatids with an extra chromosome, again leading to trisomy. The other daughter cell receives none of the sister chromatids, which can also lead to monosomy. Non-disjunction can lead to a variety of genetic disorders and conditions, depending on which chromosomes are affected. Non-disjunction is a critical concept in genetics because it results in chromosomal abnormalities and genetic disorders. It highlights the importance of proper chromosome segregation during meiosis for the normal development and health of offspring. Additionally, understanding the genetic basis of these conditions can provide insights into inheritance patterns and genetic counseling. So let's dive into some of the trisomy syndromes. So the occurrence of trisomy syndromes is typically associated with errors in the process of chromosome segregation during the formation of gametes or the early fetal development stages. The exact cause of these errors is not always known, but factors such as maternal age can influence the likelihood of trisomy. Parental testing, including amniocentesis and chronic villus sampling, CVS, can detect trisomy syndromes during pregnancy. Early diagnosis always allows for informed decision-making and the opportunity to plan for the medical and social support needed to address these specific challenges associated with these syndromes. It's very important to note that each individual with a trisomy syndrome is unique, and the severity and the combination of symptoms can vary widely. Advances in medical care and supportive services has improved the quality of life and lifespan for many individuals with these syndromes. So first, let's talk about trisomy 21, also known as Down syndrome. Trisomy 21 is the most common trisomy syndrome and occurs when there are 3 copies of chromosome 21. Down syndrome is characterized by intellectual disabilities of varying degrees, distinctive facial features such as almond eyes and flat nasal bridges, and a higher risk of congenital heart defects, gastrointestinal issues, and other health problems. Individuals with Down syndrome can lead fulfilling lives with appropriate support, early intervention, and medical care. Life expectancy has increased significantly for individuals with Down syndrome. So next, let's talk about trisomy 18 and 13, also known as Turner syndrome. Turner syndrome is also known as monosomy X. Not trisomy, monosomy X. This is a chromosomal disorder that affects females. It results from the complete or partial absence of one of the 2 X chromosomes typically present in females. Instead of the usual karyotype of 46XX, individuals with Turner syndrome have a karyotype of 45X, or a variant with structural abnormalities in one of the X chromosomes. This genetic condition can lead to a range of physical and medical features. Next, let's talk about Klinefelter syndrome. Klinefelter syndrome, also known as 47XXY syndrome. This is a chromosomal disorder that affects males. It is characterized by the presence of an extra X chromosome, resulting in a karyotype of 47 chromosomes instead of the typical 46XY. This additional X chromosome leads to a range of physical, developmental, and hormonal differences. Let me talk about some of the key features that are aspects of this syndrome. So this syndrome is typically caused by a random genetic event that occurs during the formation of sperm or egg, resulting in the presence of the extra X. Individuals with this syndrome may have subtle physical differences, including taller than average stature, longer limbs, and a tendency towards a slender build. Gynecomastia, the development of breast tissue, can occur due to hormonal imbalances. So we've already touched on this topic a little bit, but let's talk more about sex determination. Sex determination in genetics refers to the process by which an individual's biological sex is determined based on their genetic makeup. In most species, including humans, sex determination is influenced by specific sex chromosomes or the presence or absence of certain genes. Sex chromosomes, like we already said earlier in the presentation, are labeled X and Y. Females have two X's, males have an X and a Y. Sex is determined at conception when a sperm cell carrying either X or Y chromosome fertilizes an egg that's always carrying an X chromosome. Offspring inherit their genetic sex from the combination of sex chromosomes contributed by the parents. So let's talk about the sex-determining gene. The presence or absence of a specific gene on the Y chromosome, called the SRY gene, plays a central role in determining male development. The SRY gene triggers the development of male-specific reproductive structures, such as the testes. Hormones play a big role in this as well. The presence of testes in males leads to the production of male sex hormones, predominantly testosterone. These hormones drive the development of secondary sexual characteristics such as facial hair and a deeper voice. In females, the absence of the SRY gene leads to the development of ovaries and the production of female sex hormones, including estrogen and progesterone. These hormones drive the development of secondary sexual characteristics such as breast development and a wider pelvis. It's important to note that while genetic sex is determined at conception, the development of an individual's sexual characteristics is a complex process that involves the interplay of genetics, hormones, and environmental factors. Not all individuals fit neatly into the binary categories of male and female. Some may have variations in sex chromosomes or differences in sexual development. Sex determination can also vary among different species. Some species use a variety of mechanisms, including genetic, environmental, or both, to determine sex. Understanding the sex determination is crucial in genetics, reproductive biology, and medical fields, as it helps explain the basis of sexual dimorphism and the development of primary and secondary sexual characteristics in organisms. So I thought it would be cool to put a slide on hermaphroditism in animals. So hermaphroditism in animals refers to the genetic condition when an individual possesses both male and female reproductive organs. This adaptation offers several benefits. So number one would be increased reproductive success. Hermaphrodites can mate with any available individual, making it easier to find a mate and reproduce. This flexibility enhances reproductive success. Number two would be efficient fertilization. Hermaphrodites can self-fertilize in some cases, ensuring reproductive success even in isolation or when suitable mates are scarce. Reduce competition. Hermaphroditism reduces competition for mates, as individuals can mate with each other reciprocally, enhancing cooperation within a population. And number four, resource utilization. Hermaphrodites can allocate their energy and resources more efficiently between male and female functions, adapting to changing environmental conditions. Genetically, hermaphroditism often involves the presence of both male and female reproductive tissues, allowing the individual to engage in both self-fertilization and cross-fertilization, depending on circumstances. This genetic adaptation contributes to the survival and the reproduction of hermaphroditic animals in various ecological niches. Hermaphroditic animals, sometimes referred to as sequential hermaphrodites, have the unique capability of possessing both female and male reproductive organs, as we talked about in the last slide. This adaptation can be advantageous in various ecological and reproductive contexts. So here are some examples of a couple of these animals. So earthworms. Many species of earthworms are hermaphroditic. When two earthworms mate, they can exchange sperm with each other, allowing both individuals to fertilize their eggs. Slugs and snails. Some gastropods, including certain species of slugs and snails, are hermaphroditic. They can exchange sperm with each other during mating. Clownfish. So, clownfish, like the popular character in Nemo, are sequential hermaphrodites. They live in social groups dominated by a single breeding pair. If the female dies, the dominant male will change sex and become the new female. And I hope I don't butcher this one, but let's say anemone fish. Okay, this group includes clownfish, as mentioned earlier. Like other members of the damselfish family, anemone fish can change from male to female when the dominant female of the social group dies. Now let's talk about traits. X-linked and Y-linked traits are two types of genetic traits that are inherited on the X and Y chromosomes. These chromosomes determine an individual's sex, like we've talked about, with females having X and males having XY. So let's talk about the differences between X and Y-linked traits. So on this slide, we'll cover X-linked traits. So number one would be inheritance. X-linked traits are caused by genes located on the X chromosome. These traits can be either recessive or dominant. So in females, the presence of two X chromosomes allow for the expression of both dominant and recessive linked traits. In males, there's only one X chromosome. Therefore, any X-linked trait will be expressed, whether it is dominant or recessive. There is no second X chromosome to carry a different allele. So let's talk about the transmission from parents. Mothers can pass X-linked traits to both sons and daughters because they contribute one X chromosome to their offspring. Fathers can only pass X-linked traits to their daughters because they contribute a Y chromosome to their sons. Examples of some X-linked traits can be hemophilia, color blindness, including red and green color blindness, and muscle dystrophy. So now let's talk about our Y-linked traits. Y-linked traits are caused by the genes located on the Y chromosome. These traits are specific to males because only males possess a Y chromosome. Y-linked traits are exclusively expressed in males XY because females lack a Y chromosome. Y-linked traits are passed from fathers to their sons. Sons inherit their Y chromosome from their fathers. Some examples of Y-linked traits are, well, they're relatively rare, and there are fewer known Y-linked traits compared to X-linked traits. So Y-linked traits can include genetic markers used in paternity testing. So to summarize, X-linked traits are found on the X chromosome and can be expressed in both males and females, although their expression pattern differs. Y-linked traits are located on the Y chromosome and are exclusive to males, as only males carry the Y chromosome. Understanding the inheritance patterns and expressions of X-linked and Y-linked traits is essential in genetics. It can be relevant in the study of genetic disorders and inheritance patterns.